Hello and welcome everyone to our second day of conference. Today it's all about research. I am Orly Marquardt. I am the Vice President of NTSAD, and most importantly, I am William's mom. William was diagnosed in 2007 at 18 months of age with GM1 gangliosidosis and died from this disease in 2014 at the age of 8. Upon receiving the diagnosis and prognosis of GM1, my husband Rod and I, like most parents, immediately sought out information regarding any hope for treatments that would stop this disease from progressing. We were fortunate to have a network of friends and family who were in the medical and research fields who could also assist us. It was not long before we realized that there were no existing treatment protocols for GM1. It was through this network of friends who helped us search for treatment options that we were advised to connect with NTSAD. In April of 2007, we attended our first family conference in Boston, and it was here where we found our community, who quickly became our family. NTSAD became a lifeline for our family in many ways. At the conference, we attended the research session where a presentation was given about the possibility of using gene therapy as a treatment option for GM1. The evidence at this point lied within positive outcomes in mice models. Having a science background, my husband and I both knew that it would be unlikely that an effective treatment would be developed in time to save our son. However, there was hope, and NTSAD provided us a platform to be part of the effort to find a cure. We started by participating in the natural history study for GM1. By providing William's health information, we hoped that it would help the researchers better understand GM1 so that they could develop effective treatments. Through the Day of Hope started by NTSAD in 2011, our daughter, who was 9 at the time, joined the research efforts by hosting a lemonade stand to raise awareness and much needed funds. In the years that followed, she continued this effort through hosting various events and was proud to share her brother's story with others and raise money for research. Through our engagement, I have realized the power of the family voice in advancing research. As families, we are able to share significant details of the disease with doctors, researchers, and industry, details that cannot be found in textbooks. This knowledge helped inform areas of study and leads to more patient-centered innovations. The knowledge about Tay-Sachs, GM1, Sandhoff, and Canavan diseases have come a long way since my first research session in 2007. As board VP, I was involved in the strategic planning work started 2 years ago by NTSAD in response to these research advancements and the evolving needs of our families. We updated our mission statement to say, NTSAD leads the worldwide fight to treat and cure Tay-Sachs, Canavan, GM1, and Sandhoff diseases by driving research, forging collaboration, and fostering community. Supporting families is the center of everything we do. So what does this mission statement mean for our goals and work with families? We have 3 strategic initiatives. On the left, you have building community, which is what this conference is all about. And on the right, you see that we want to provide families with the resources and expertise that they need. Today, we are focused on the middle goal, which is all about research. We connect families, researchers, clinicians, industry, and other stakeholders to advance research and develop treatments. To move forward, we talked about with a lot of experts to gather their inputs. And we also set up task forces with some of these experts to summarize the current status of research, identify knowledge gaps, and provide recommendations. The overall goal is to inform how NTSAD should spend money and effort to impact research and improve patients' lives in the future. When NTSAD first started funding research in 2002, we were trying to start and to help advance more research in this field because there was so little funding in Canavan, GM1, Sandhoff, and Tay-Sachs diseases. Everything has changed. as you can see with the increasing industry involvement and clinical trials. Now we can and should be more selective regarding research investments. Therefore, one of the task forces we set up looked at updating our view of what's needed in basic and translational research. Another focus of the task force is with clinical development. Here, the principle of ensuring that the voice of the patient is at the forefront so that drug developers truly understand what is important to the patient. Because our diseases are rare and there's so much variability, we also learned that bringing all the small pockets of data and information together and sharing it is a value that we should embrace. We can also make progress faster by learning lessons from other gene-based diseases. While carrier screening has figured prominently in NTSAD history and legacy, now the focus has turned to other types of testing, newborn screening, as well as earlier diagnosis for symptomatic patients. Figuring out early detection will be key to better clinical care and successful treatments. This leads to the focus areas for two other task forces. Lastly, we understood that to succeed in this work, we need to develop more internal expertise and also be able to be more fully engaged in this work. Therefore, NTSAD is recruiting a brand new position, a research director. With this resource, NTSAD can strengthen its contributions to clinical trial, strategy, study design, and outcome measures. The position is posted on our website, and I'm just saying that perhaps our next research director is in this Zoom room. Now, I would like to move forward and share a video with you that features the leaders of our task forces, Doctors Fran Platt, Florian Eichler, Joan Koitzer, and Jerry Cox. We are very grateful to them and the many others who were involved in this initiative. Following the video, Stacy Kalish will lead a panel about the importance of family participation and data in research.
Basic science and translational research. My name's Fran Platt. I'm based at the University of Oxford in the pharmacology department, and it's been my great privilege and pleasure to have been NTSAD's Scientific Advisory Committee Chair for a number of years. Now, I've been delighted to have been joined by several task force members. Several of these individuals may be familiar to you, but let me just quickly introduce them. Sandra Dazzo is based in St. Jude's in Memphis, and she's had a particular interest in the gangliosidosis, particularly GM1. Steve Walkley has worked on multiple lysosomal storage diseases over the years, including the GM2 gangliosidosis, and Doug Martin has worked with a variety of animal models of lysosomal disorders, and Rick Pryor at the NIH has also made several mouse models of lysosomal disorders. We were joined on the Canavan side of things by David Pleasure from UC Davis, who's an expert on Canavan and who's pioneering gene therapy approaches for this disease. So the purpose of the task force when we all met was to update the research areas where NTSAD can play a significant funding or other role. So this really focused on areas where NTSAD specifically could catalyse progress. So the recommendations of this task force was to enhance researchers' abilities to access patient samples. If a new researcher comes into this field, we want them to feel as though the tools to do research on these diseases are freely and easily accessible. And many people will start with patient-derived cells, and this is something that we don't yet have a complete panel of available to researchers. So this is something that we could do in a very straightforward way that would encourage more researchers to come into the field. We've also got a number of very useful animal models in this area, but we do have some deficiencies. And one of those is that we lack a large animal model for Canavan disease. And this is really essential for making the transition from trialing gene therapy, for example, in a very small mouse brain, to a species that is somewhere intermediate between mouse and human. So this was identified as one of the goals of this, the next phase of NTSAD's efforts in the research area. We also have new mouse models of GM1 and GM2 that have different pros and cons. And we're now building up a very comprehensive set of animal models for GM1, GM2 and Canavan. And so more effort on this was also highlighted as being important. We're still interested in soliciting grant proposals from the research community to help build our understanding of these diseases. And in discussing with the task force, three main areas came to light: pathophysiology at the cellular level, the effect of these gene defects on myelination, and also the effects of these mutations on driving inflammation in the brain. And we would be very interested in funding more seed grants that really will allow greater progress to be made in these three key areas. We're also very keen to fund opportunistic research proposals that we realise have come up with a new idea or a new finding. They're very difficult to predict. You never know where new advances are going to come from. And so we want to be a nimble and flexible organisation and always have an open door for opportunistic funding of new treatment approaches, which will benefit the patient community in both the short and the longer term.
Hi, my name is Florian Eichler. I'm a neurologist at Massachusetts General Hospital, and delighted to be here and speak to the clinical development task force that I led as part of this recent initiative of NTSAD. The task force members are shown here. We had a broad array of talents in this task force, ranging from clinicians and physician scientists to industry members. Tanya Fisher really brought her industry expertise to this task force, and people like Jonathan Mink and Mark Patterson, many years of clinical trial experience particularly in lysosomal diseases. Cynthia Tift, I don't need to introduce, somebody with deep knowledge and experience in the field of GM1, GM2, many other lysosomal diseases. Camille Kaur is a medical student who I had the pleasure of mentoring a few years ago at Massachusetts General Hospital, and joined us from the University of Rochester. And Brian Wishart, a physiatrist who thinks a lot about symptom management and was helping us think through the clinical needs of patients on a day-to-day basis. So why have a task force on clinical development, and what does that actually mean? We are currently at an exciting time in history where certain treatments and therapies are coming to the fore, and we are now confronted with questions around clinical trial design, what kinds of outcome measures, what kinds of biomarkers to measure in order to know whether the treatments we are using are actually successful. And so the NTSAD Clinical Development Initiative was really aiming to understand where are areas that represent gaps here that NTSAD should help close. Are there partnerships with or collaborations with third parties that are needed? and what kinds of organizations and forums of experts are needed to help close gaps and move things forward. So, a long story short, what kinds of recommendations did we come up with for NTSAD? We found a paucity of disease-specific severity scales, and this is certainly an area that the various rare diseases that NTSAD advocates for could help in, by recognizing that also different phenotypes within the individual diseases have different needs, different stages of disease, where there are different needs. So in infantile onset disease, it could be quite different in terms of scale development than a late onset disease stage, where often there is more of a chronic course where you might need more granular measures that are addressed today with wearables. So thinking about the nature of different stages of disease and different phenotypes seems to be a focus that NTSAD could help with in the coming years. Symptom management and understanding how to guide families is another area where we don't have really publications for health professionals and families. And this is an area where if we brought physiatrists or physical therapists, occupational therapists together, we could really do a lot for families on a day-to-day basis. Obviously, NTSAD participating in funding these above initiatives where needed could make a big difference.
Hi, everyone. I'm Joan Koitzer. I ran the Newborn Screening Task Force, and it's a pleasure to share a summary of our work with you. Newborn screening is without a doubt the best way to guarantee that every child with the disease has a chance to be treated early. It is testing every baby born in a state for a panel of life-threatening diseases. The goal of newborn screening is to identify and treat every affected child as soon as possible before irreversible symptoms can develop. The panel of diseases included in newborn screening is determined by each state, but that panel is recommended federally. The panel is called the Recommended Uniform Screening Panel, or the RUSP. Newborn screening is part of every state's public health system. It is not a diagnostic test. It's a screening test, and every abnormal test result results in a newborn being referred for confirmatory diagnostic testing. And every diagnosed child is monitored and referred for treatment when they need it. In general, for a disease to be included in a newborn screening panel, the disease needs to be able to be detected biochemically before it's clinically obvious. The incidence of disease needs to be high enough to make newborn screening worthwhile. And in general, that's 1 in 15,000 to 1 in 60,000 births. The benefits of early diagnosis and intervention must have been demonstrated previously before disease gets put on a newborn screening panel, and a simple, inexpensive test that identifies patients must be available. So our recommendation is that we prepare each disease nomination for inclusion in the RUSP, that we make sure that we finish the R&D on the Tay-Sachs enzyme assay and the Canavan substrate assays, that we plan newborn screening pilot studies, and we approach both private and public agencies for funding, that we complete the RUS nomination form for each disease and update it periodically so that we know when they'll be ready. And I think the most important recommendation that came out of this is that NTSAD should establish and coordinate a collaborative working group for each of these diseases. That includes all stakeholders, so patients, patient advocacy groups, newborn screening experts, physicians, scientists, and industry, to work together to get these diseases on the RUSP as soon as possible. And I would like to just update this by saying that recognizing that there will be efficiencies among the 4 diseases, on April 8th, we already had a first meeting with some physicians, some industry, some patient advocacy groups, and newborn screening experts. And we have formed a collaborative working group to promote newborn screening for these diseases. Thank you.
Hello, my name is Jerry Cox, and I'll be sharing with you the results from the Early Symptomatic Diagnosis Task Force. By way of introduction, I'm a NTSAD board member, and I've had a long career in industry developing drugs for rare diseases. I'm also a practicing clinical geneticist at Boston Children's Hospital. So today I'm going to describe the outcome of our task force on early symptomatic diagnosis. And these are all the people who contributed to our findings, starting with Karen Grinzade, who's a fellow board member and genetic counselor, Simone Azevedo, who's a healthcare marketing and patient advocacy specialist, Heather Lau, who's a neurologist and clinical trialist, Dale Mussey, who's a molecular geneticist that works at a diagnostic lab, and Emily Gaudet, who's a genetic counseling intern who did a lot of the computer searches for us regarding genetic testing laboratories. So our task force really sought to understand where NTSAD could bring together our collective perspectives to try to understand how to arrive at earlier diagnoses through changes to the current way that genetic testing is being performed, as well as raising awareness among patients and families, as well as the healthcare specialists and PCPs that see them. So our recommendations to NTSAD to try to reduce this time gap was as follows. Before seeing the physician, we believe that educating patients and families through storytelling on social media, either internet, magazines, newspapers, with stories that would resonate with families that are not yet diagnosed. And in addition, raising awareness among PCPs, early intervention programs that would see the young children, and then some of the therapists like speech and occupational and physical therapists for the older patients, about the importance of referring patients to a neurologist or geneticist for a diagnostic evaluation. And then once within the medical system, reducing that diagnostic odyssey by educating specialists about the rare diseases and the benefits of timely diagnosis. Importantly, advocating with the labs to include the NTSAD diseases on their relevant gene panels. We don't think that there'll be objections to this. I think they just haven't thought of all these diseases to include. And then finally, there is a trend towards this new test called whole exome sequencing, which looks at all the different genes. But it's important that the labs know what the signs and symptoms of the patient are as they look at the different genes. And so having them be aware of the different NTSAD diseases as they do the whole exome sequencing will be very important as well. So over the past year, we've concluded the 4 task forces. We have a series of recommendations, and now we need to keep the momentum going to implement these recommendations. And thank you very much, and enjoy the rest of the conference.
Hi, everyone. Thank you to all of our task force participants and task force leaders for sharing your experience and your time with us and helping NTSAD determine the direction of our research efforts. You can see that we've set an ambitious agenda for the organization. Because of this, and based on these recommendations, as Aurelie mentioned, we're already working to recruit a research director who can help us advance these important initiatives. A link to the job posting is on our website. I'm Stacy Kalish, and if we haven't met, I'm a longtime NTSAD community member, a medical geneticist, and the current NTSAD Board of Directors president. I can't advance. There we go. Okay. So today we're going to talk about ways patients and families can be involved in and can impact research, focusing specifically on data patients can provide to researchers. As you just heard from one of our task force speakers, Dr. Eichler, NTSAD and our medical and scientific advisors have identified this as a priority because this patient-provided data is so important for assessing potentially effective therapies. This is distinct from being in a clinical trial with an intervention, and it instead involves patients and families directly providing their data for the sake of research. We've got two speakers today, Dr. Caitlin Nichols and Dr. Cynthia Tift, who will discuss some of the research they've been working on in partnerships with patients and families. We'll also have a discussion together where our speakers will answer your questions. You can type them in the Q&A box on your screen as you think of them. The Q&A box should be at the bottom of your screen, and it's probably the second button in from the left. When we were considering this topic, Dr. Nichols explained it well in this way. Patients and patient data can bridge the gap between basic research, the work that's happening at benches and labs, and clinical trials, where novel interventions are studied in actual patients. Some of you may have participated in this type of research before, and some of you may be actively participating in this type of research now, or maybe considering it for your future. There are essentially three ways that patients and families can provide their data to aid research, and we'll talk about each of these in a bit more detail in a minute. Patients and families can share their data directly through surveys and questionnaires. Patients and families can also provide their medical records or access to their medical records for review by the research team. And finally, patients can present themselves physically for evaluations and assessments to provide information. This is research that patients physically participate in, but does not involve an intervention such as a medication. In each of these categories, there are a number of ways that patients and families can participate, and NTSAD patients and families have and are currently participating in a number of these. So again, we'll be talking about these three ways patients and families can share data and participate in research. These here show examples of ways patients and families with our diseases, with Canavan disease, GM1 gangliosidosis, Sandhoff disease, and Tay-Sachs disease, have or are currently helping researchers gather data. Again, the first way for patients and families to participate in providing data is through direct providing of data through surveys and questionnaires. Here are examples of these. Patient Insight Networks, or PINs, exist for Canavan disease, for GM1 gangliosidosis, and for GM2 gangliosidosis, or Tay-Sachs and Sandhoff diseases together. In the middle here shows examples of some of the data that can be collected from the infantile GM2 PIN, showing that there are 32 patients or families who have participated, showing some of their demographic information and some of their experiences in obtaining a diagnosis and receiving medical care. For example, in this pie graph, you can see how many of the participants have participated in types of diagnostic testing, such as blood tests, MRIs, EEGs, and genetic testing. Registries collect specific information about those living with the disease. The CATS Foundation, which some of you may be familiar with, has a registry working with the European Tay-Sachs and Sandhoff Charity Consortium, which collects patient and family entered data, including sociodemographic and disease-related characteristics, such as variant of disease, age at diagnosis, symptomology, and genetic mutations. The CATS Foundation has a poster available for viewing in the exhibit hall with data from the infantile patients in their registry. Patients and families can also participate in focus groups and patient preference studies, where small groups of patients or families gather with researchers for discussion of how diseases impact individuals and families. This includes a caregiver preference study getting started for GM1 gangliosidosis, now beginning recruitment, led by NTSAD, Cure GM1 Foundation, and RTI International. This study will help define what parents feel are the most meaningful symptoms to address when developing therapies for children. The study will collect actual parent perspectives, which will be shared collectively with families, the FDA, pharmaceutical companies, and doctors studying GM1 so that they can better incorporate the patient voice into research and drug development. There will be more information available about this study shared in the breakout group for GM1 and after the conference. Surveys can also be used to collect patient data. These are called retrospective natural history studies, and they can allow physicians and scientists to learn about the natural history of diseases directly from family members. This paper on the right of the screen shows a study that was published in the Journal of Pediatrics in 2011 that described the natural history of infantile GM2 based on surveys completed by NTSAD parents. This allowed families and their children to contribute to research even when their children were no longer living. Another way that patients can provide data to researchers is through video assessments, such as those the Casimir study for GM1 have collected. As we said, the second way families can impact research is by providing copies of their medical records or access to their medical records. This can be used to build natural history retrospectively through review of medical records, or it can be used by researchers to seek insights into obtaining a diagnosis and living with a disease. This is what All Stripes does, gaining insights into where patients are living with disease and what their experiences are. There are currently All Stripes studies ongoing for GM1 and GM2, and this is some of what can be learned here on this slide. Dr. Caitlin Nichols will speak more about this in a minute. Another way to use patient medical records to learn new information is by meta-analysis of the data already in the medical literature. In this type of study, researchers review all of the available published literature and combine it to be able to draw more accurate conclusions from the larger amount of data. These initial studies are often case reports of single or few patients, literature that is describing patient diagnoses and clinical features. And then finally, the last way patients can impact research is by really providing themselves for assessments and for evaluations. This can include traveling to research sites for prospective natural history studies, looking forward rather than backwards, and looking at how disease impacts those living with it. This includes natural history studies for our diseases, like the one done for GM1 at the NIH, the ones ongoing for GM1 and GM2 at the University of Minnesota, as well as the CAN-INFORM study ongoing for Canavan disease. This can also include the late onset clinical assessments that occurred and many of our community participated in at previous conferences. Dr. Cynthia Tift will speak about some of her work with both the GM1 natural history study and the late onset GM2 assessments in a moment. Patients can also provide data by participating in remote studies by wearing devices that can collect data about their movements and allow for patients to submit data directly to researchers as they're living with disease. And finally, patients and families, including sometimes carrier parents and carrier and even non-carrier siblings, can impact research by providing samples of fluids like blood, urine, and spinal fluid, or by providing other tissues such as skin samples. A number of our community, including many siblings, participated in a study a few years ago at a conference by providing a drop of blood. As a Tay-Sachs disease carrier, I was able to participate in this too. So I'll put this up here once more as a reminder of all the different ways that patients and families can impact research. And now I'd like to introduce our speakers. Dr. Caitlin Nichols is the research director at All Stripes, a medical data science company with the goal of accelerating treatments for people affected by rare diseases. Through her work, she designs research studies for industry, academic, and patient group collaborators, and plans how to communicate these findings into the broader scientific community. And many of you already know Dr. Cynthia Tift, but for those of you who don't, Dr. Tift is a medical geneticist at the NIH, where she is the deputy clinical director. Her clinical and research interests are in lysosomal diseases. She has been involved in research into the natural history of GM1 and late onset GM2, which she'll talk about today, and is the principal investigator for the juvenile GM1 gene therapy clinical trial ongoing at the NIH. Dr. Tift is also a longstanding member of the NTSAD Scientific Advisory Committee, or SAC, and is a former SAC chair. She has attended many NTSAD conferences and events. Dr. Nichols, I will turn it over to you.
Thank you so much for the introduction. It's really such a pleasure to be with you all today. One of my favorite things about working with the rare disease community at All Stripes is getting to speak with patients, families, and other advocates. So I'm very excited to talk with you today about what Stacy mentioned about how medical records, either yours or those of your loved ones, can help to power research. So what can we learn from medical records? A lot of times, as was mentioned, we might think about research as happening in the lab at a bench with someone with a white coat, or perhaps participating in a clinical trial. But there's so much that we can learn that can bridge that gap from digging back into a patient's medical history. So we can understand things from even before patients were diagnosed, through their diagnostic journey, and then how they're being treated now and what the outcomes of those treatments are. So for example, we can look at the earliest symptoms and when those symptoms occur, and it's really helpful to understand more fully what patients' symptoms are, because then drug companies and the FDA can start to understand what are the symptoms that our drug should even address in order for it to be considered effective and to really help patients and their families. In terms of the diagnostic journey, we can understand things like what does it take for patients to get from that initial symptom to actually receiving an accurate diagnosis? What kinds of doctors are they seeing in the healthcare system? What kind of tests are they having? And these questions are important because if we can figure out ways that we could identify patients earlier, then we might be able to help clinicians get them a faster diagnosis. And then looking at things into after a patient has already been diagnosed, how are they being treated out in the real world in clinics all over the country or all over the world? What are the outcomes of those different types of treatments or interventions? And this can be helpful because if we can see that perhaps one intervention is helpful for a specific subset of patients, then we can know to try to help get those patients that particular intervention. So where does All Stripes come in? We are a medical data science company, as was mentioned, with the goal of unlocking new treatments for people affected by rare disease. So we work with stakeholders across the rare disease community, from patients, families, doctors, advocacy groups, drug companies, academic scientists, to help find treatments for patients like John, who's one of our GM2 ambassadors, and for other rare patients like him. So how does this process work? Well, what we do as a company, as you can see here on the right of the screen, is that we'll create a disease-specific landing page. Oh, sorry, there's a fire truck. All right, we create a disease-specific landing page for each condition that we host on the platform. You can see here is our platform for GM1. And so patients, parents, or caregivers can create an All Stripes account. It's worth noting that former caregivers can also create accounts for patients that have passed on. And then the users can sign an umbrella research consent, as well as a medical record authorization form that allows us to go and collect medical records from all their facilities on their behalf, as well as a list of facilities where the patients have received care. And so then our operations team will go and collect all those records, digitize them, and upload them to a secure platform where the patient or their family can access them. They can share them with a clinician who they'd like to be involved in their care, or a caregiver as well. And then they can use that to help coordinate their care. It's worth noting that participants always own their data. They can delete their account or download it and take it to another study at any time. And then participants who have opted in to participate in research are regularly notified about milestones in the community as well as research studies that we're participating in. And so the umbrella research consent that I mentioned is for a variety of different studies that would allow us to dig back into the medical records to pull out de-identified data. We then structure that de-identified data and work with researchers at academic institutions or at pharmaceutical companies to advance rare disease research and drug development in that condition. So how do we use the de-identified patient data of patients who'd like to participate? One instance here is this program that we call the All Stripes Insights program. This is showing data from our GM1 cohort, which we were so proud to launch with Cure GM1 and have NTSAD join on shortly after. So through outreach to the community from both of these amazing organizations, we were able to have 28 consented research participants who had allowed us to dig into their records to understand more about GM1. So for each condition on the platform, we will produce this report that's really a core level of data that we view as a research launchpad that the communities can take out to academic investigator X or pharma company Y and say, here's these patients who are ready and willing to participate in research. Let's see what we can learn. What more can we learn from their medical records? We look at a variety of data points, where the patients are, what types of records and types of facilities the patients are visiting, digging more into the symptoms, the diagnosis, genetics when that's relevant, as well as how the patients are being managed, what types of healthcare resources they're using out in the real world. So I just wanted to talk briefly about a quick example. This is from our GM1 cohort showing common symptoms that patients with GM1 experience. And you can see that the most common ones in our cohort were hypotonia or low muscle tone, as well as seizures, followed by swallowing difficulties, orthopedic issues like kyphosis and scoliosis, rounding of the spine, drooling and choking, as well as some ophthalmological issues. But we also know that just seeing a bar chart like this it doesn't really speak to the full impact of the condition on the patients and families who are living with the disease. And so along with creating this research launchpad that we hope will spark more research, we want to return these insights back to the patients and families to be used for their own education and advocacy efforts. So you can see here, taking that bar chart into something that's accessible to the community, not just to illustrate the prevalence of a symptom, but also to show what is the impact of this symptom on patients and families who are living with GM1. What's the effect of having seizures in your child, etc. We can also share these insights back to the scientific community to help inform ongoing studies. So this is an example of a poster we presented this year at the World Conference. I believe you can see it in the poster gallery. And this was highlighting data that we collected in GM1, again, with partnership with Cure GM1 and NTSAD. And then finally, we can use these data to power in-depth research studies. I was really excited to hear the update about wanting to add the NTSAD conditions to the RUSP, because we're involved in another effort with some other foundations to add a condition called Niemann-Pick disease type C, hopefully to the RUSP. So we work with foundations, with governmental organizations, for example, the National Center for Advancing Translational Sciences at NIH, pharmaceutical companies, and academic and nonprofit organizations to advance research that will hopefully help to improve diagnosis, help to improve policy, as well as to further drug development. And so those of you who are participating in All Stripes, sorry, my dog is in the background. Those of you who are All Stripes participants who may have seen this news from NTSAD, we have a GM2 community, as was mentioned, and we, with the community, reached the recruitment milestone last month, which was really exciting. So congratulations to the community there. We're so proud to partner with an amazing advocacy organization like NTSAD, and thinking about what medical records can do to power research. As I was looking into, what are the goals of NTSAD, this longstanding organization, our goals are the same: to help advance education, support for patients and families, research, and ultimately treatments. And so we hope that these efforts will contribute to all of those different points. Thank you so much for having me. As I mentioned, we have GM1 and GM2 cohorts, but we know that those are not the only conditions that NTSAD covers. And so we'd love to explore potentially launching other conditions with NTSAD as well. And I'll be happy to take any questions during the discussion period. Thank you.
Good afternoon. My name is Cynthia Tiff, for those of you who don't know me, and I'm thrilled to be here at the next annual family conference. Certainly wish we were here, we were all together, but I will tell you, for those of you, I can't see myself, but for those of you who can see me, yes, it is the third year of purple hair, but it doesn't come out very well on Zoom. So today I want to talk about more generally about what kinds of things families can provide, the key information they can provide on information on prospective natural history studies. And by that prospective, I mean beginning at a point in which you enroll in the study and then continuing on over the time that we follow you, as opposed to a retrospective study where we would be going back and either looking at your medical records or asking you to complete a questionnaire. And there are some differences, but there are also some challenges. So the things that I would like to talk about today briefly are: what is a natural history study? What are the advantages of participating? What are the disadvantages or limitations? How is my or my child's data used? And who's going to benefit from participation in a natural history study? So what is a natural history study? It is, as I said, a way of evaluating a group of patients. And in this sense, most prospective natural history studies are much smaller than reviews of medical records or questionnaire retrospective studies, because they involve a lot more time and energy on the part of investigators and on the part of the patients who participate. So there are people that have the same disorder or the same subset of disorder, and basically we use the same set of metrics to look at the same kinds of things in each patient. Evaluations are based on initially what we know about the condition, and then we try to expand our knowledge about the condition by studying patients carefully. So one important goal is to better understand the disease, to possibly identify where we could apply treatments that might be effective in treating the condition. Another aim is to identify outcome measures or things we can measure once we design a clinical trial. And so I call these measures of what do you call success? And I think that's a lot of what we're doing in our natural history studies. So what are the advantages? So participating in a natural history study often involves a very comprehensive evaluation by specialists who have seen many other folks with your disorder. In rare diseases, that's particularly important. For the diseases I study, GM1 and GM2, where the incidence is 1 in tens of thousands. It's a way of having experienced set of eyes that have seen many people with the same condition. As I say, this is particularly important if the disorder is rare or in some cases ultra rare. Researchers collect the same information on each participant. That's called cross-sectional collection of data, each participant at different stages in their disease. And we often follow participants over many years. So we have not only a snapshot of each individual in time, but actually are able to look at the way their disease changes over time. And we know that's particularly important for our diseases because they tend to be progressive, some more quickly than others, but even for the adult onset diseases, they are slowly progressive, as we know. Researchers may learn things about a condition that were previously unknown. We call that expanding the phenotype and our ability to be able to understand things that were not previously known about the disease. And so another advantage is that participants may learn more about their disorder. They may learn better how to manage it effectively. We may be able to provide, if not a definitive therapy, at least recommendations for additional types of therapy that may improve their quality of life. For example, dietary information on special services or physical therapy or speech therapy that might be helpful, assistive devices, for example. Those might be some examples of supportive therapy that would not be definitive treatment, but might improve participants' quality of life. So what are the disadvantages of participating in a natural history study? It requires time. It requires a time commitment. At the NIH, we generally bring our participants in once a year, once every 2 years, for several days at a time. So time lost from work or school is certainly a consideration when you're trying to decide whether or not even to participate in a natural history study. Participants may need to travel to the evaluation site. Some assessments have inherent associated risks. For example, the pediatric patients in our trial that we perform MRI studies on undergo sedation in order for us to complete those studies. And as everyone knows, there's always a risk to putting a child to sleep. The risks have to balance the benefits, as our institutional review board will tell you, but there may be some associated risk. Studies are costly to conduct, natural history studies, so they are rarely funded by research grants. So it takes a unique situation to be able to do natural history studies because they're so costly. The NIH has been, the intramural NIH has been one place that's been possible, and the National Human Genome Research Institute has a number of natural history studies ongoing for families with rare diseases. Participation is voluntary. This is like a clinical trial in the sense that the risks and the benefits for each part of the study need to be explained to participants before they start, so they can really make an informed decision as to whether or not they want to enroll in the study in the first place. In our case, we usually send the consent form to families before they even plan to come. And then, of course, we go over it very carefully with them once they arrive. Participants and parents are able to ask questions, raise concerns about the protocol. If they agree with that, they would then sign a consent form that's witnessed by myself or another member of the team. And it's important to know that in natural history studies, just like in clinical trials, participants can withdraw their consent at any point, and they can also opt out for certain portions of the study that they just don't feel comfortable with. And lastly, the principal investigator, usually the clinician, can decide that a participant is not suitable for the study, and we can decline to enroll them on that basis. For example, if we consider them too medically fragile to be able to complete the assessments that we need in the study, and that it would be more risky than the typical patient to be able to participate. So the NIH gangliosidosis study, and this is a study that's been going for more than 10 years, looks at patients with GM1 and patients with GM2. They can be infantile, juvenile, and adult, but I can tell you that the two buckets of patients that we have seen more of, I think, and are more common in the population have been GM1 gangliosidosis patients with late infantile and juvenile disease, and Tay-Sachs patients with late onset disease. And I often tell patients when they come for the study, there's good news and there's bad news, and it's both the same. The good news is you're going to get about a year's worth of evaluation in a week. And the bad news is that you're going to get about a year's worth of evaluation in a week, and you're going to be really tired at the end of the week. Some of the things we might look at in a patient when they come in, besides a very complete history and physical, might be an ultrasound looking for enlargement of liver and spleen, an echocardiogram looking at the structure of the heart, a neurologist assessment, blood draws, a skin biopsy, as Stacy was talking about, in order to obtain cells that can be grown in culture for researchers to test new treatments. We might do an EEG, we might test hearing and vision. We might do imaging studies, we might do x-rays on bones for the patients who have skeletal disease. So you see, it's a vast array of sort of looking at multiple organ systems to try and arrive at a good picture of the disorder. So how do we use this data? When sufficient patients have been recruited and conclusions can be drawn from that group of patients, the results are generally published in the medical literature in an attempt to educate other practitioners, other physicians about the disorder. It's particularly important for our diseases. And Joan Kreutzer was mentioning about the task force for newborn screening. You really have to understand the natural history of the disease in order to begin to think about newborn screening, to begin to think about early diagnosis. And we really want to educate practitioners in the community about our diseases. This is just a screenshot of a couple of our most recent publications. The one on the right here, this mHealth, Stacy also referred to, this was the wearable technologies that some of our adult onset patients helped us with contributing data. And the one on the left is regarding the bony changes in GM1. Study data, once it's published, once the data is published, we make all the raw data available in a de-identified format for others to benefit from the information. That could be other academics, that could be companies, that could be the advocacy community. But the data is made available at the time that we publish it. Data from our natural history studies can be used, as I said, to identify outcome measures for trials of new therapies, no matter what those therapies might be. And important for our perspective is that the natural history studies performed here at the NIH, the data from those can't be sold. Once published, this data must become freely available because it was generated using federal monies. Who will benefit from my child's participation in a natural history study? Well, a lot of us, the researcher and the study team, because we are able to publish the results of our work, the medical community, because they're able to learn more about our diseases, the participants, because we're able to give them pointers about the evaluations, the comprehensive studies we've done, and give them information on how to best manage their disease and improve their quality of life. And the patient community, like NTSAD, because that really informs all of us about our diseases and how best to study them. So with that, I would like to thank all our study participants and their families, as well as NTSAD, for giving me the chance to talk about this. And I, in the panel, like Caitlin, would be happy to answer your questions.
Great. Thank you so much, Dr. Nichols and Dr. Tift, for these presentations. So again, I'm going to invite our audience to type questions for our panelists using the Q&A button on your screen. The button should be at the bottom of your screen. While we wait for the questions to come in, I'll start with a question of my own. So many families have raised multiple times and in different ways concerns about privacy in sharing their records or their loved ones' records. So how can researchers or companies, can they share data without asking the patients who share it with them? Can they sell data to others? And how do researchers and companies keep data safe and confidential?
I'll be happy to start. I think that the privacy concern is so important. We know that medical data is precious and it should be treated with the utmost respect. So as I mentioned, we will only collect records if a patient consents for us to do so, and patients can opt into research or opt out of research at any time. If a patient opts out of research, then we'll stop using their records in research. So it's always, participants always own their data, and that is number one. It's our number one priority to protect patients' data and to keep it safe. But it is also important to note that anytime you share information, especially electronically, that there's a small chance that something could go awry there. So we always want to inform participants that there is a risk when you consent to participate in research or when you share records. But again, we have an internal data security team who follows all the best practices, and we're all trained on how to protect information that we work with. In terms of the model for how we as a company make money, what we do is we sell pharma our services to collect and analyze the data. But again, we don't sell patient data because patients always own their data. And if we're always transparent about who we're working with. So for whatever reason, you're like, no, we don't want you to work with that pharma company, you can totally opt out of the program, and then your data won't be included in our research. But if there's any follow-ups, I'd be happy to take those as well.
And Dr. Tiff, do you want to address sort of patient data security or in the academic setting?
Yeah, I would say in the academic setting, in medical centers and at the NIH, patient records that under clinical trials are treated in exactly the same way your medical records are treated when you go to your physician or your hospital and your primary care provider. They are your medical records. They cannot be sent to anyone else without your permission, and certainly can't be sent with identifiers without your permission. Now, the first thing we would do in our clinical trial is we assign you a de-identified number. We call it a GSL number. And that's the way we keep track of you in our databases. Certainly the medical establishment at the NIH knows, just like every hospital knows your name and your records, but those medical records are protected in the same way all of your medical records are protected. When we use the research data, it's all done and everything is stored. For example, if we collect samples, all those samples are stored by your de-identified number, not by your name.
Great, thank you. So another question came in from a family who has participated in a natural history study, but has a question about if you're currently enrolled in a natural history study, and I think this is for a prospective study, how would this work with regards to collecting or submitting ongoing medical records? So I think this is someone who participated and has submitted previous medical records, but now has ongoing procedures, hospital stays, other types of medical interventions, and wants to know, if you submitted medical records at the beginning of your study, is there a mechanism to continue to submit them as you go forward?
Sure. We're
Yes.
always, this is a partnership. I should emphasize, this is a partnership between participants and ourselves. And, you inform us, we inform you, and we say to all of our patients, if there's something that's significant that happens in your medical history, we would love to hear about it. By the same token, if you have a question of us between visits and just want to know, just want a question answered or us to think more deeply about what you're going through, we'll be happy to do that as well. Now, often, to be honest with you, we do this by email, but my general, and so we can send medical security emails. My general feeling is that if you are emailing about your child, you are giving me permission to email you about your child. But I don't usually initiate those with your child or with you in an unsecure way, I guess I should say.
I'll add, for us, we also do collect records in an ongoing fashion. So we'll typically refresh a patient's records every year. So if there are new facilities, new procedures, things like that, then those will be captured.
Great. So another question that came in was about the publication of data, which I know Dr. Tift and Dr. Nichols both mentioned in your presentations. And the question is specifically that the presentation of data can take a long time. So are there other ways, and what other ways can you share data in the interim? Or while you think about that, can you address sort of some of the frustration and ways that, that we can help get this important information being useful in a faster way?
I mean, we can certainly share information about each patient with that participant and that participant's family. And certainly at that point, then they're willing to, they're available to share it to anyone else. You're right. It takes a very long time to accumulate enough data, because what you're looking to do is not just look at an individual patient. You're looking to get enough patient data to be able to draw a conclusion that is a reliable conclusion, or that, in medical statistical ease is clinically significant. There is a significant change from point A to point B, whether it is disease progression of, neurodevelopmental testing or skeletal things, or, you have to accumulate a certain amount of information in order to be able to call it a difference, to be sure that that's what's actually happening. Otherwise, you're publishing something that is incorrect. And although that happens sometimes, you'd like for it not to be able to happen. So, and in the interim, it's hard because if we don't have enough information to draw a conclusion, then I'm not sure anyone else is going to have enough information to draw a conclusion. Unless we pool data. I mean, I guess there would be instances where if another institution would have been collecting exactly the same data under the same conditions, it would be possible to pool data. I can tell you that is what happened between the NTSAD, when we did the studies in the late onset Tay-Sachs adults during NTSAD family meetings. And really that was done with Sanofi under Genzyme, now Sanofi's guidance, and under their IRB. Well, at one point they really needed additional data on certain metrics that we had been collecting as part of our natural history study. And under an agreement with Sanofi, we actually provided that data to them so they could decide whether or not they had anything to move forward with a clinical trial. So sometimes we can do it that way.
Yeah. I want to echo what Dr. Tiff said about really communicating and sharing what we're finding, and with the caveat that this is not, you know, the capital T truth. The scientific community hasn't necessarily evaluated these interim findings, but when we go to conferences and share interim findings or at clinical roundtables, I've been in some really interesting discussions where people are saying, are you seeing that? Because I'm seeing this too, and maybe this should spark a new idea. So I think really communicating and collaborating with everyone in the community can be helpful. And then certainly wanting to return back insights to individual patients. That's something we do in our platform, is that patients can actually see, for example, where do I sit in, you know, the distribution of when a patient had their first symptom. Here's the whole range and here's where I sit. Things like that we can return in the interim while we're doing the work of cleaning and analyzing and making sure that we're making really robust conclusions for an eventual publication.
Great. So there was another question about collaboration that I think was really focusing on collaboration in terms of prospective natural history studies. So this is from a family whose experience has been participating in what sounds like two different prospective natural history studies. And it's sort of an acknowledgement of the time that it takes to participate these, that there is certainly strain on the families and on the children when they're pediatric patients, and in terms of duplicating testing that's involved in these prospective studies, and thinking about collaboration of the sharing of data sort of during this process rather than after the data is collected to help lead to better outcomes and less duplication.
There certainly are, through, I guess what's now part of NCATS, consortia for different diseases. And Stacy, I'm sure you know this well, that there may not be any one center that has a large number of patients, but maybe 4 or 5 centers together will form a consortium. I'm pulling this out of the air, let's say for Williams syndrome or something like that. And each institution will agree on a set of metrics that they're going to measure in each of their patients. And so often those collaboratives are started before, you know, as the project is coming along, as it's sort of beginning, and each academic institution decides on what it can do, about how many patients it will be able to recruit from its geographic area, and what metrics are going to be looked at. And then often there is even a coordinating center that will collect all the data together, aggregate it, and then it will be published collaboratively by each center.
Yeah. One challenge I think that we see in the rare disease community is this duplication of data, and there's an increasing push for wanting to have a unique identifier so that there's a de-identified code that goes with each patient. And so then you can know, you know, I'm in Dr. Tiff's study, but I'm in this other study, but it's the same patient. But this is tricky based on regulations, as well as getting people to agree on what's the best system so we can protect patient privacy while also making sure that those identifiers really are going to be specific to the person. So there's a number of different stakeholders in the rare community that are trying to hash this out, and hopefully we'll start to see some progress, but that definitely is a challenge, and can appreciate the time and effort that goes into trying to participate in so many different research efforts, for sure.
Great. Well, thank you. I think with that, we'll need to end the panel discussion. For anyone who's typed in questions that we didn't have time to get to, we do have a record of the questions and we will seek to get answers afterwards. So I'd like to thank Dr. Caitlin Nichols and Dr. Cynthia Tift for this great conversation about the importance of patient data and the many ways that patients and families can participate in research and provide this crucial data. We'd like you to also hear from some families about why they've chosen to participate in research and share their data in this video.
NTSAD families have been driving research for nearly 7 decades through advocacy and fundraising. But perhaps families' greatest legacy is their ability to advance research through their active participation, whether it be through natural history studies, data collection and surveys, patient focus groups, FDA listening sessions, and now clinical trials. As we work towards a world where rare diseases are no longer fatal or debilitating, families hold the keys to unlocking the door to effective treatments for themselves and future generations. Families' participation in research is invaluable. Everything we do is in support of families and to find effective treatments.
If our son was going to have to go through such a horrible disease, that we wanted him to have as many opportunities as possible to contribute something positive. And so that translated for us into research, because in that way he could help probably not himself as much, but other kids.
I think that the majority of us are doing it to help those adolescents and infants to hopefully extend their lives if possible. If we can give the drug companies or the hospitals or the researchers, if we can expose some magic serum or, God willing, we are doing it for them more than, I would say, even more so than for ourselves.
If anybody can learn anything about how to cure a disease or how to make a child comfortable while they struggle with some of the more severe symptoms of a neurogenetic disease is really important.
Being a part of it made me feel like I was contributing to a cure. I never felt I was being exploited. They weren't trying to get anything from us. All they wanted to do was learn about the disease.
We had probably gone maybe 3 times to NIH when the doctor there sat down with us and just told us really honestly that a cure for this disease would happen, but it was going to be a long time. It probably wouldn't be when our son was able to utilize it. You can imagine that kind of hits you like a ton of bricks to realize that what you're doing really isn't for you, but it is for other people. We just felt like that was important for our son, that he needed to have, that his life has significance. And it has significance on a daily basis, and we appreciate that. But it also has significance in the long term, because we know that he's going to be a part of finding a cure for this.
It was important for me to participate in research like this because it was my little bit that I could do to help push for that cure. Because Finnegan was a whole lot. He wasn't sleeping. I was sleep deprived for a solid year. This was something I could do real quick, but meaningful. And so it made me feel like I wasn't just sitting back and doing nothing to fix this for him or for other future children. I'm sick of seeing children die. Sick of seeing it.
We've given blood samples, we've given urine samples, we've given tissue samples. We had to think a lot about the spinal fluid because it is a little bit more invasive test, but we chose to do that also. We've done a lot of x-rays that they've used for comparison factors with other children. A lot of things that sometimes seem kind of repetitive, but they really aren't in a disease that's progressive, because they are looking at and documenting over time what happens. And it's so important for them to know that. That's called the natural history of the disease. And they really can't come up with treatments or figure out how to gauge if those treatments are working unless they know what the natural history of the disease is.
I went to Houston a couple of years ago and went to a focus group that was for parents that had lost children of Canavan disease. And it was, sort of like I like to say, like an oral history of the signs that you noticed before your child was diagnosed, the progression of his disease, what was helpful in accommodations that you made to make him comfortable. And some of the things that I've learned along the way with taking care of my son were from other parents. It's a wonderful way to share your child's story. And it's also, for me personally, a way to connect with Ben's memory and also be able to help other children that will have genetic diseases in the future.
When Aparito had their wearable tracking devices that tracked our strides, that tracked our blood pressure, things like that we would actually throughout the day, we would self-report several times. And there was a collection of data that Aparito provided researchers with that was, I think, very helpful in some regard.
First of all, you just have to look for the opportunities, and that comes from being involved in organizations as much as you can and reading newsletters and just things that might pop up on the internet related to your disease. And then there have been small things that we've been able to do to contributing samples that we've shipped across the country. Just look for those opportunities as a family, and then also wait for things to drop into your lap and be ready to say yes. Even if it's just a little bit out of your comfort zone, if you can push yourself to be a part of that. And one of the reasons is just because this disease is so rare. If we don't all participate, then how are they ever going to come up with a cure for something so rare? I don't ever regret anything that we've done or contributed, but you do have to be ready. Ready for the unexpected. Isn't that how we all live every day? Just kind of ready for the unexpected.
So I was able to do something to help find a cure for Tay-Sachs, but on the same side of it, the 3 other parents that were involved in my group are the 3 people that I'm the closest to now. The relationship that we built, because you're asking each other questions and you're responding to each other. I learned about the disease as I was sharing our experiences.
It may not have a big impact on what happens for our child, but we're really committed to contributing to research for other children. In the long run, when I look back on it, it's been a good and positive thing for the emotions in our family, and not just for myself or my son, but for our marriage and for our other children. I mean, they had to make some sacrifices. I think that that's been good for them. I feel like they have more compassion for other children, other families.
I'm completely hopeful that there will be a cure for other neurogenetic diseases. And I think we're pretty much, looking at the progress that people are making with research, and hopefully now with increased funding coming along, that there will be families that when their child is told they have Tay-Sachs or Canavan disease, it would be like, okay, so what's the treatment? And they will be able to be told that there is one. So yes, I'm very hopeful in the future that there will be.
I think it's the most exciting time that it's ever been to be part of NTSAD or to be joining this journey, that we're closer than we've ever been to clinical trials, including having some ongoing, closer than we've ever been to having approved therapies, closer than we've ever been to having earlier diagnosis. And so I think it's a really exciting time. Hi, everyone. Just going to wrap up. Let me put those up again. Okay. Hopefully you're seeing all of this. So thanks to NTSAD Dad Dan Redfield for creating that video. Just going to let you know what's going to happen for the rest of the afternoon. So after this, we'll have a short break. And then at 2 o'clock Eastern time, so just about 35 minutes, we will start our breakout groups. Just to note that you'll have to leave this session, and then you'll have to rejoin with a new link. There'll be a link for each of the breakout groups, and that came in the email that you received today with all of today's links. Please don't sign on before about 1:55 so that we can get everything reorganized. So we'll have a breakout group for each of our diseases. The Canavan group will include topics on antisense therapy, also on Canavan and the brain. There'll be information about a natural history clinical trial, the CAN-INFORM study, and about ongoing clinical trials or soon to be starting clinical trials from ASPA Therapeutics and Cure Rare Disease. The GM1 group will include potential therapies in development from Gain Therapeutics, Idorsia, and Azafaros, and information on clinical trials in progress from Lysogene, Passage Bio, and Cytogene therapies. There'll be a group for infantile and juvenile GM2 or Sandhoff Tay-Sachs disease, and this will include information on potential therapies in development from Idorsia, from Azafaros, and from Recursion. And then also on clinical trials in progress from Cytogene therapies and from Taysha gene therapies. And then finally, we'll have a group for late onset GM2, and this will include updates on work from the LOTS think tank, updates on gene therapy in sheep, clinical updates on medications, questions about clinical trials, and updates on clinical trials in progress from IntraBio and from Sanofi Genzyme. You can really see how far we've come with this wide variety of research from early science, patient-provided natural history data, preclinical work, and clinical trials. I'd also like to let you know that we will have an online exhibit hall that has abstracts and posters from some of these studies and from other studies. And this is also includes some abstracts and posters from work that was funded by the NTSAD Research Initiative. The link for the exhibit hall is available on our website as well. So before I close the Research Day kickoff, I'd like to thank our sponsors for supporting the annual Family Conference. Thank you to Sanofi Genzyme. Thank you to Aspa Therapeutics, Recursion, oop, that went fast, Laura and Simeon Schindelman, and Taysha Gene Therapies. And thank you to all of our sponsors for sustaining our important work. Your support makes everything that we do possible. And then with that, I'll put these up once more as you leave so that you can see the topics for the breakout groups. We hope to see everyone there. Thank you.